Well, hello, everybody. Thanks for sticking with me. So we're getting there. Just two more lectures to go, two more topics to cover. So today's topic is essentially looking at some very important techniques that are used in modern genetics laboratories to sort of look at genetics from a whole genome perspective and to look at protein function in the same way. So we're going to begin with this first mini lecture where we talk about some functional genomics that people are currently doing, followed up with some proteomics. And then we're going to finish this topic in the next mini lecture by talking about bioinformatics. So to begin with, let's talk about functional genomics and proteomics. So what are these techniques that we're going to talk about? As I said, these kind of techniques, functional genomics, proteomics, and bioinformatics, are designed to get at what is going on in a cell when you consider the whole genome, not just individual genes or proteins. So functional genomics is looking at exactly that, the sort of roles of the genetic sequence in the life of a species. And mostly this is, of course, understanding gene function, but it can also be understanding the regulation of gene expression that we talked about previously in the class. Proteomics is sort of just looking at proteins, not DNA sequence itself. And bioinformatics is essentially a very broad term that refers to using computers to analyze genetic and biological information, which is something that's sort of exploded in the last decade as we're generating way more DNA sequencing data and proteomics data. So we're going to begin by talking about my computer would function, functional genomics. So we have the ability now, and we've had the ability for about 10 years really, to sequence a lot of DNA very quickly using technologies that are called next generation or next gen sequencing technologies. When I was in graduate school, for example, NIH sequenced the Drosophila genome, which was the model organism that I worked on. And it was the first sort of big genome of a model organism to be sequenced. And we all thought at the time, oh my God, am I going to be out of a job now? Because we know the whole sequence of the whole genome. This was a whole new world. But what we learned is that whilst that is useful for lots of different reasons, understanding how the genome functions is way more complicated than just sequencing the DNA to begin with. And some would argue, myself included, that it is more interesting to look at the function of a genome than just the sequencing of that genome. So now we can sort of do a bunch of genomic sequencing and look at many genes and their function at the same time in a given cell or tissue. And the way we can do that is using DNA microarrays, which is the first technique or technology that we're going to talk about. And DNA microarrays are sort of cool because they allow us to see what genes are expressed and at what levels in any given cell at any given time. They're called DNA microarrays because they are literally a tiny array of DNA sequences. This slide that you see on this slide, I guess. So this microscope slide that you're looking at, I use this particular picture because you can see somebody's fingers and see how big it is. Those little squares that you see on it, each one of those squares contains thousands of little tiny dots of DNA just dotted onto the slide. Kind of one way to do it is with something very similar to an inkjet printer. This is a relatively small DNA microarray, but there are still 30,000 dots of DNA on that slide. So the way to think about DNA microarrays is a piece of glass with tiny dots of DNA all over it that are sort of microscopic. And very importantly, which DNA sequence is present in each one of those spots is known. So you can represent basically every gene in the genome as a single dot of DNA. And what you can use those to do is you can sort of use them as probes to see which genes are transcribed and the level of gene transcription that occurs in a cell. So how do you do that? So here's how you use a DNA microarray to study gene expression. So this is shown in, of course, totally different scales so that we can understand what's happening. So what's shown in the top right-hand corner there is a glass slide. This time it's just got 6 spots on it, but you could imagine that as 30,000 dots or 100,000 dots. Conceptually, it's the same idea, right? So we have 6 different spots of DNA, A through F. A is in the top left-hand corner of the slide, F is in the bottom right-hand corner, and we know what sequence is present on each one of those dots, and that sequence is the sequence of a specific gene in the genome. And what I can then do is, if I have some, for example, some liver cells growing in a dish, right, and I want to ask what genes are expressed in those liver cells and at what levels, I can do that. So what I do is isolate the messenger RNA from those cells, from those liver cells, because, you know, whatever messenger RNA I find in those cells, it's from genes that are being expressed in those cells. So here we have a mixture of mRNA that I got from those cells. As you can see, there are 3 different types represented, A, D, and F, and there are more of certain types than other types, right? Actually, I think here there are 3 copies of A, 3 copies of F, and 3 copies of D. Then what I can do is something called a reverse transcription experiment, which you just basically take the mRNAs and you can convert them back into DNA because it's very stable. So that's a good thing to do. And in this process, you also fluorescently label the nucleic acid so that it glows green under when you stimulate it with a certain wavelength of light. So we've taken our mRNA, which, remember, is going to represent which genes are transcribed, and the more mRNA that's present, the higher transcription level of that gene. In this particular example, right, there's no expression of B, C, or E in this particular cell. And then you basically take that single-stranded DNA that you just made and you hybridize it to the single-stranded DNA that's on the slide. As you guys know, if 2 sequences, single-stranded pieces of DNA are complementary, they will hybridize with each other because they will just base pair, essentially. And by doing this, you can sort of wash the slide with these fluorescently labeled DNA molecules, and they will base pair with their corresponding gene. So in this case, A, D, and F are all lighting up because the fluorescently labeled DNA has matched to those spots. The more mRNA that was present in the beginning, the more cDNA will be made for that gene, and the brighter the spot will glow, indicating a higher level of expression of that gene in those cells. And so this is a very powerful technique because we can take some cells and we can do experiments on them, throw a hormone on them or something, and see how gene expression changes when you stimulate the cells in some way. But the important thing to recognize is that you can basically look at every single gene that's being expressed in the cells at the same time, which is a very powerful technique. If you thought about this, you could think of there's many different things you can do. So this table just sort of tells you a few of those. I want to just point out one that's particularly interesting, tumor profiling in the middle there. You can actually isolate cells from, say, for example, I had a pancreatic tumor. You could isolate my regular pancreatic cells that are not part of the tumor, and you could look at do a DNA microarray experiment on those. And then you could take some cells from my tumor and do a microarray experiment on those and see what the differences are in gene expression between my normal cells and my tumor cells. And that can help basically diagnose certain types of tumors and maybe suggest treatments for those particular cancers. So DNA microarrays, very powerful, whole genome. I look at the whole genome levels of gene expression at the same time. A very similar technique is called RNA-Seq, which is slightly newer. And it's kind of similar. This time, we're looking at RNA levels in a given cell, which allows you to compare what are called transcriptomes. So the genome is all of your DNA sequence. Your transcriptome is what genes are being expressed in any given cell at any given time. And you can do very similar experiments with RNA-Seq that you do with DNA microarrays. You just can look at differences in expression when you compare two different cells. Starts off very similarly. You take a bunch of, this is a technique of RNA-Seq, you take a bunch of mRNA from some cells. In this case, we've got 3 different types, green, blue, and red. You can see there's way more green than blue and red. So here's our mRNAs, and we're going to break them into small fragments just to make this experiment a little easier to do. Then we're going to do a very similar thing to what we did with the DNA microarray, which is we're going to make complementary DNA, which is just using reverse transcriptase to reverse transcribe the mRNA into DNA. That DNA is called complementary DNA, or cDNA for short. And then we're going to sequence those RNAs so that we can identify the different genes that are expressed. And then we will, after we've sequenced them, we will figure out how many green sequences were there, how many red sequences were there, how many blue sequences were there. And then using a computer, we will align them to the entire genome sequence. And what you can see here is that we have gene A, which is green, gene B, which is red, gene C, which is blue. Those 2 dotted lines along the bottom are meant to represent the sequence of those genes in the genome. And when we align all the sequences that we just sequenced, then we can see there's way more green sequences present than red ones, for example. And that tells us there were way more green transcripts present than red transcripts. So the green gene, gene A, was being expressed at a higher level than the red gene, gene B. And if you go back and look at the origins of the experiment, you'll see that's true. There's way more green transcripts at the top of the experiment. And so this is another way that you can measure expression levels of every gene in the genome at the same time. Now, these types of experiments often lead to, identification of certain genes that are particularly interesting. So in this experiment, maybe gene A, all of a sudden you're like, whoa, this gene that I'd never heard of is expressed at a really high level in this cell. Maybe it's really important. So how do you then figure that out? Well, the best way to figure out what a gene does is to delete it from the genome and see if it has any effect, if it has any phenotype. That used to be a big pain in the butt to do, because you'd find a gene that you were interested in and you'd have to knock it out yourself. Making a gene knockout is just deleting a gene from the genome. And so to facilitate that speed of research, the scientific community is making gene knockout collections. So as you guys are probably aware, there are a relatively limited number of model organisms that scientists study. Drosophila, the one that I used to study, mice, C. elegans. You've looked at some of these in the lab portion of this class. For each of those model organisms, there are basically collections of gene knockouts so that you can order any gene knockout that you want. So I work on fruit flies. I find gene A that I'm interested in. I can get some flies sent back to me from a collection that have that gene knocked out, and then I can find out what it does. They're currently doing this. It's already done for things like yeast, E. coli, fruit flies, and it's underway for mice as well. That gives us some idea of what a gene might do in an organism. And then proteomics is essentially a way to figure out what proteins are doing at the molecular level. So experiments like DNA microarrays or RNA-Seq might lead you to be particularly interested in a gene. But what you really want to do when you've identified a gene that you're interested in is ask what the protein is doing. Because if you really want to know what a gene is doing, right, you have to study what the little molecular machine, the protein, is doing. So anything that you figure out, any genes that you identify using any of these fancy techniques, you need to look at what the protein is doing to really figure out what the role of that protein is. And the reason that you have to do that is because genes don't tell you the whole story. So the proteome, again, I'm sure you're getting the hang of this terminology. We live in the world of ohms these days. So the entire collection of proteins in a cell is called its proteome. And the proteome of any particular cell is actually not completely predicted from its genome. So one gene can make different types of proteins. And that kind of makes sense. Human beings only have about 22,000 genes, but we have way more proteins than that because you can make more than one protein from any given gene. Here are some of the ways that can occur. Alternative splicing is one way. You guys should have learned about this in cell bio, but essentially, as you know, in higher organisms, in eukaryotes, genes are split into introns and exons, the exons being the part that codes for the DNA. And you get splicing that occurs after transcription to splice out the introns and leave you with just the exons. And one of the cool things that has been discovered about eukaryotic genomes is that one gene can undergo different types of splicing to mix together different exons. That is called alternative splicing. So here you can see an example of that. We have a gene with 6 exons. When it's transcribed initially, the immature or pre-mRNA, which is shown at the top here, has blue introns, and then the different colored exons. And different ways of splicing can effectively get different exons together. So the first alternatively spliced mRNA is giving us exons 1, 2, 4, 5, and 6, but not exon 3. The second version is exon 1, 3, 4, 5, and 6, but not exon 2. And the third example is 1, 2, 4, and 6. So that one just has 4 exons. It's lacking 3 and 5. And each one of those different messenger RNAs that's been alternatively spliced is obviously going to give you a slightly different protein when it's translated. And that's one way that you can have different, more proteins than you have genes in your genome. That is the most common way that I'm aware of that you can get one gene to produce multiple proteins. There are a couple of others. There are cases of RNA editing, which is pretty rare, but that can actually lead to different a different sequence in the message than there was in the DNA. So that's a little crazy. It's very non-Mendelian in its process. And then the last one doesn't really have anything to do with the sequence of an mRNA or indeed a protein. But proteins can be covalently modified after they've been translated to change their function. You guys learned a bit about this when we were talking about transcription factors, that they can be phosphorylated, for example. A phosphate group can get stuck on them, and that changes their function. And there's lots of other instances of things like that. Proteins they can have little things called ubiquitin moieties stuck onto them, phosphates, they can be methylated, and these post-translational covalent modifications can affect protein function. Now, because of that, scientists wanted a way to say, hey, if I have some cells like liver cells, for example, it would be really cool to be able to ask what proteins are present, not just what transcripts are present, but what proteins are present, and are they post-translationally modified? And so there is a way you can do that. The way that I'm going to show you is called two-dimensional gel electrophoresis. And it basically allows me to take some cells and to separate out all the proteins and analyze them individually. So I can take my liver cells, I can lyse them to get all of the proteins out, and then I can subject them to 2D or two-dimensional gel electrophoresis. It's called that because it literally is that. It's gel electrophoresis in 2 dimensions. So the first step, I run my proteins through a gel. This is a very special kind of gel. It's called an isoelectric focusing gel. And it basically, it's one way to separate proteins based on their pH. And so the dark purple lines that you see are meant to be bands that were separated using this isoelectric focusing technique. Then that's the first dimension of gel electrophoresis. And then what I do is I take that first gel and I literally turn it around and lay it flat on top of another gel, which is an SDS page gel, which is one of the things that you did in Cell Bio Lab. And so then this separates the proteins based on size. And so we have 2 different ways of separating proteins. One is their pH, basically, and the second one is by their size. So using this technique, you can sort of tease out proteins so that each different black spot on this gel should represent a single protein. And when you actually look at a gel that's been performed like this, it looks like this kind of ugly thing that you see on the right, where there's lots of different black spots. Each one of those black spots should be one protein, and you can literally cut those out of this gel and get the protein sequence from those spots on the gel. This is something that I've actually done, and it works pretty good. So you can then cut out the spots that you want. You can send them to another lab, and they will sequence those spots. And again, similarly to RNA-Seq or a DNA microarray, you can do this with 2 different cell types, or a cell, cancer cells versus regular cells. Look if there is any different proteins in one cell versus the other cell. You cut out the spot that you're interested in, and then you do something called mass spec or mass spectrometry, which at least when I did it, involved cutting out a little spot from that gel, mailing it to a company, and then they email you a file back that says, hey, this spot equals this amino acid sequence, which is pretty neat, because once you get an amino acid sequence, you can figure out what gene you're talking about, because we'll understand how the genetic code is used, and you can figure out what gene sequence is going to code for that protein. Interestingly too, mass spectrometry can tell you whether your protein is phosphorylated, for example, or methylated. And so it gives you some information about potential post-translational modifications as well. Pretty cool. Okay, last but not least, I'm going to talk about a more challenging type of experiment. You may be asking yourself, well, hey, Dr. Parker, you can do DNA microarrays, and you said we're interested in proteins. Can you do protein microarrays? And the answer is yes. It's a little bit more challenging because proteins are not as stable as nucleic acids, and they're a lot more complicated because they have to be three-dimensional in nature. And the idea behind a protein microarray is that if I could array all of the proteins on a slide, I could take a cell lysate, I could bust my cells open, wash my proteins over the slide and see which ones interact with different proteins on that protein microarray. And that might give me some idea of, is this transcription factor talking to RNA polymerase? Does it bind to RNA polymerase? That kind of thing. Those are very technically difficult experiments. But again, of course, you can think through a lot of particularly cool applications that these protein microarrays could have. For example, pharmacology, the one at the bottom there, if we think about human health, you could have all of the human proteins on a microarray, put some fluorescently labeled drugs over it, and see which proteins they bind to, which could give you potentially some drug targets for those particular drugs. Okay, so those are some various ways that you can look at the genome, the proteome, and the transcriptome of cells at a whole level, and compare one cell type versus another cell type to ask some questions, what genes are expressed and at what levels. And that can give you a lot of information on how these cells function. So that's it for this lecture. We will move on to the next one. I hope you're all doing okay.